Hi and welcome to Biostat Squid. This is part 2 of my series on single-cell RNA-seq integration methods. So if you haven't watched part 1 yet, you can do that first to get an overview of the methods we're going to be comparing today. So if you're ready, let's dive in. Okay, so previously we presented different integration tools that follow different strategies to integrate data from different batches. And as I mentioned, integration methods essentially try to correct batch effects whilst preserving as much biological signal as possible. So now that it's time to integrate your data, how do you know which method is best? Essentially, we want to know how each method scores on how well it preserves biological signal and how well it removes the technical effects. Now, to compare methods more objectively, several benchmarking studies have been published. Most of the content of this video is based on the Luken et al. Nature Methods paper from 2022. So Malte Luken and colleagues did an excellent job at benchmarking 16 popular integration methods across different integration tasks. Their study provides a more objective guide on which method works best under different scenarios, which is great because, as you will see, different tools perform best with different integration challenges. So the first thing is to define these integration challenges or integration tasks. Each integration task represents a different source of variation or batch effect you'd like to correct for. Are you trying to integrate data from different institutes? Are you doing a cross-species comparison? Do you have data from different tissue sites of the same patient? Your first step is to identify what are your batch effects, as that will help you focus on the benchmarking results for that integration task. In the Luken paper, the main integration tasks included simple datasets with clear cell types, like pancreatic cells from different studies, or the human immune task, which includes different tissue sites. Here, the challenge is to integrate datasets from different batches, which may have been processed at different times, or by different labs or people, or they may come from different tissue sites. But we expect very similar cell types, and all genes are, in this case, human genes. They all come from the same species, in any case. Then we have complex datasets with nested batch effects, like the mouse brain data with 1 million cells. Nested batch effects occur when there are multiple levels of unwanted technical variation in data, such as when samples from different experiments are processed in different batches, and those same experiments are also organized into different lab groups. They also tested integration methods with data with strong biological variations, so different species or tissues or disease states. The authors found that the most challenging batch effects across the integration tasks were due to species, sampling locations, and single nucleus versus single cell data. So, for example, the human and mouse immune datasets gets a bit trickier because we're integrating data from 2 different species. So even the same cell types have different gene expression profiles, with some genes being species specific, and there will also be unique cell types to a species. So it's a bit more difficult to integrate. And finally, we have different data modalities when we're trying to integrate single-cell RNA-seq and single-cell ATAC-seq, for example. So the reason I'm mentioning all of this is that one of the key takeaways of this publication is that method performance is dependent on the complexity of the integration task. So depending on what effects you want to correct for, one method would be better at correcting it than another. For example, Harmony works really well on simple integration tasks, but on more complex integration tasks, it doesn't really rank top 3. There, you might be better off choosing a method like Scanorama with embeddings or SCVI. But we'll get into the results in just a minute. Now, how do you know if your cells are well integrated? So the next step is to figure out how exactly do we measure whether a specific integration method performs better than another. So a good integration method should merge and transform the data in a way that it removes the batch effects between the different data sets whilst keeping as much as the real biological signal as possible. Note that a big part of how well we can do this computationally is tied to how the experiment was designed. So, for example, if we want to test for differences between blue and pink lymphocytes represented by stars, if we process all blue samples in one batch and all pink samples in another batch, it means batch and condition or color is confounded. It's going to be very difficult to correct for batch differences, so purely technical differences, and see the actual biological differences between blue and pink stars or lymphocytes. If we don't correct, we won't be sure whether the differences we're seeing are due to the batch effects, so technical differences between both batches. And if we do apply a correction method, it might overcorrect the data. So we get a good overlap between batches, but we're losing the biological signal. In this simple example, we cannot distinguish stars from circles anymore. So a good experimental design will enable you to correct for batch effects, but keep these biological signals. In this case, stars versus circles. So the biological signals you want to observe. All this is to say that batch correction methods are a great bioinformatic tool to have, but they cannot do miracles. And often if you don't use them properly, they can actually be harmful for the analysis you want to do. But okay, that was just a little side note. Back to our integration methods and how to measure their success. So how do we quantify technical effect removal and keeping biological signals? So the researchers used 14 metrics divided into two categories. First, batch effect removal metrics, which assess how well the methods mix cells from different batches. You don't need to understand how each of these metrics works to be able to choose a good integration method for your data. But here's a brief overview of what they do. So you have k-nearest neighbour batch effect test, which checks if a cell neighbours come from multiple batches. Graph integration local inverse Simpson's index, which measures diversity of batches in cell neighbourhoods. Then we have ASW, which measures how separated batches are. Graph connectivity, which checks if cells with the same identity are connected across batches. And principal components regression, which measures variance explained by batch. Next, we have biological conservation metrics, which assess how well methods preserve real biological differences. And here we can distinguish two categories, depending whether we know or not the cell types already. Label conservation metrics harness the known labels or cell types to measure how well they are clustering together. Basically, we know that tumour cells should cluster together with tumour cells from other samples or batches. So, similar to batch effect correction metrics, we have different ways of measuring this clustering at cell type level. We have the normalized mutual information and adjusted RAND index, which basically compare clustering results to known cell types. Then we have cell type ASW, which measures how well cell types are separated. We have graph clustering local inverse Simpson's index, which measures how well cells of the same type cluster together. And then we have isolated label scores, which assess preservation of rare cell types. On the other hand, we have label-free conservation metrics, which go beyond cell type labels. So you don't need to know your cell types to be able to compute these metrics. So we have cell cycle conservation, which basically assesses how the integration method preserves cell cycle variation. We have highly variable genes conservation, which basically checks if the integration method is retaining informative genes after integration. And trajectory conservation, to see if it's maintaining developmental or differentiation paths. So essentially for each method and in each integration task, we'll get a score for every single metric we mentioned. So if you're interested in a feature, you can always look at specific scores for one of these metrics. For example, if it's very important for you to retain cell cycle variation, then you might want to use the top scoring method for the cell cycle conservation metric. If you're interested in maintaining developmental or differentiation paths, then you would look for the methods with the highest scores for trajectory conservation. Otherwise, if you want to focus on the overall score of the method for a particular task, the authors also report an overall score which summarizes all this by giving 60% weight to the biological conservation metrics and 40% weight to the batch correction metrics. This basically reflects that preserving biology is more important than removing batch effects. Nice, so there's a reason why I present this as a weighing scale, and it is that a key point of the benchmarking study is that there's an inherent trade-off between removing batch effects and preserving biological variation, meaning we can't get the very best of both worlds. So more aggressive batch correction methods like BBKNN, Saravi3 or Liger do a great job at mixing cells from different batches, and they're great for getting a broad overview. However, because we're being more aggressive with the batch correction, there's a higher chance we're removing real biological differences. So these methods are more prone to merging different cell states, especially if there's very little difference between them. If we take the opposite, so more conservative methods like DESC or CONOS, we find that they're better at preserving nuanced biological variation and retaining rare cell types, but of course they might leave residual batch structure, meaning cells from different batches might not fully integrate. For example, you might see a clear separation between M1 and M2 macrophages in two distinct clusters, but when we look closely, we realize that within each group, they're not really integrated well and they are separating by sample. Then there are also more balanced methods like maybe Scanorama, SCVI, or FastMNN, which achieve a good compromise between both objectives and work well across diverse scenarios. But before you go and completely discard the more conservative or more aggressive integration methods, you need to understand that sometimes they might be the right choice for your data too. If you have strong, obvious batch effects that need correction, so for example cross-species comparison, or you just want a high-level overview of major cell types, then a more aggressive batch removal method might be right for you. And if you're dealing with subtle cell states or rare populations or developmental trajectories, or even if biological variation might be confounded with batch effects, then you might want a more conservative approach, knowing that some batch effects will remain. Figure 3 in the paper shows a very nice scatter plot of the mean overall batch correction score against the mean overall bioconservation score for a few selected methods on RNA tasks. Ideally, you want a method that's in the top right corner, meaning it scores highly both for batch correction and bioconservation. But as you can see from the error bars, these scores change depending on the task. For example, if we zoom in on CONOS, it might perform really well in terms of conserving the biology for some tasks, but poorly for others. So again, you'll get a better estimate of how each method will perform on your data if you check the stats for a similar integration task. We'll see how to do this in just a bit. Nice. So as a recap, we've talked about different integration tasks, different methods we can use to overcome these integration challenges, and we've also mentioned the ways we can measure how well each method performed. Now, if you've already used any integration tool, you might already know that there are different ways to run the tools. Many have different parameters you can optimize. So this publication focused on two main ones, using scaled or unscaled data, highly variable genes, or the full gene set. The general trend that this benchmark study observed was that most methods improved when highly variable genes were selected before integration. So in general, they got better overall scores than when you used all genes. And scaling, or Z-score normalization, showed better batch removal but worse biological conservation. So the very, very general recommendation, and again, it's better to check for specific examples that align with your data, but the general recommendation is to use highly variable genes before integrating and to avoid scaling unless batch effects are very strong. Nice. So now we're ready to go back to our original question. How do I choose an integration method for my data? So let's say you have an integration task at hand. You have several data sets you'd like to integrate. How do you decide? Well, I would recommend you to check out figure 5, which is a fantastic table the authors put together where they present general guidelines to choose an integration method. So first we have the input, so depending on if you're more used to R or Python and if you have cell type labels. So Scanvi and SEGen need cell type labels for integration, so you have to have annotated your cells first. Then they highlighted methods based on the scores across all the integration tasks. So we have top performers for smaller or simpler tasks like Scanorama, FastMNN with embedding, SCGen and Harmony. And then top performers on larger or more complex tasks like Scanvi, SCVI, Scanorama and SCGen. And for ATAC-seq integration jobs, we have Harmony and Liger. Then you can check out more specific task-related details, like which one is the best for very strong batch effects, which one is the best at distinguishing biological and technical differences, which one is best for trajectories or for datasets with different compositions. So this section of the table is very useful if you have a specific issue or integration task at hand. So apart from the expected performance we've already talked about, you also want to know whether your method is scalable, especially for tasks where you want to integrate a large number of datasets. With single cell, datasets can become really huge, so you might need more computational power, or you might be interested in faster methods or more memory efficient methods. Some methods have GPU support, for example. So the authors also scored their usability, meaning how well they are documented, if there's tutorials and active development for the tool. And finally, the output is also very important. If you're looking to do specific downstream analyses like trajectory inference, then you need corrected gene expression values. So methods that output a graph won't be adequate. Nice. So let's put all of this theory into practice with a specific example. Let's say we have a simple integration task where we want to see whether there are differences in immune cells of mice that have had the flu versus healthy controls. So we have several donors of mice in each group, control and condition. We take blood samples, and because of whatever reason, maybe time constraints or maybe the sequencers can only take a certain number of samples at a time, but we sequence them in 2 batches. When we process our single cell data and get our UMAP, we see clusters of different immune cells as we expect. Great, we can now move on to DGA analysis. But wait a minute, let's color these cells by batch. We see a clear separation of samples by batch, so the data is clearly not well integrated. We can also color them by donor, meaning the actual mouse the sample came from. We can also observe sample-specific clusters. Now, it can be okay to have sample-specific clusters if, for example, a specific subtype of patients has a rare cell type population, or maybe the viral infection in this case caused an increase in plasma cells, which are not present in the control samples or are present in negligible numbers. But in general, we want our cells clustering by cell type and not by sample. We cannot compare cell types between control and healthy samples because we wouldn't be sure whether we're observing true biological differences or differences due to the fact that some samples were sequenced in one batch versus another or come from one mouse versus another. Based on this UMAP, we would need to improve the integration a bit. Finally, we can also color them by condition. Now here we're not expecting a full integration because it's actually the condition we're investigating. So some cell types might be better integrated than others, and the UMAP might already give you some clues as to which cell types are most affected or more different as a result of the virus infection. But it doesn't need to be a perfectly integrated UMAP. Later on, when we do DGA analysis or other downstream analyses, we'll find out what's different between healthy samples and controls. But to be able to compare those groups, we first need to make sure we're not observing differences based on the actual sample they came from or the batch. So what do we do now? Well, we need to integrate our data. To choose an integration method, we can go to our benchmarking study. The setup we have, so 2 batches with different donors, is different to the human pancreas task, which comprises 6 human pancreas datasets, so same species, same tissue, and just different donors or datasets. So we're going to focus on the benchmarking results for that integration task in particular. The authors created some very nice summary tables. In the supplementary material, we can find the one for the pancreas dataset. So from their benchmarking results, we can see that Harmony and Serraz, RPCA and CCA performed best overall, followed closely by FastMNN and SCGen. Make sure to also check out the preprocessing options. We can see that it's best to use highly variable genes, and scaling works okay in this case for most of the top scorers. If our dataset has any particularities, for example, if we have particular interest in cell cycle conservation, then we might want to check out the particular score for that metric. But otherwise, you can focus on the overall score as well as the scores for batch correction and bioconservation. Interestingly, for this task, some methods perform even worse than not doing anything at all. That's why it's important to choose your integration method carefully. So again, these are the results for a very simple integration task, but if you have more complex scenarios, then check out the integration task that's more similar to your own. For example, if you have data from different tissue sites, then the human immune task is for you. Or if you want to integrate cross-species, then check out the human and mouse integration task results. Not only can you check the summary tables in the publication itself, but the authors also provided a fantastic website to explore the results in a bit more of an interactive way, which can really help you compare different methods. So you can literally click on particular methods you're interested in, or particular datasets or tasks you would like to know the stats of. So let's explore in detail the scores for the human-mouse dataset. We can see that the scores are based on 23 batches with almost 100,000 cells, with SCGen using highly variable genes and unscaled data having one of the best bioconservation and batch correction trade-offs. You can also have a pretty table where you can order by particular columns, or you can also see the scores for individual metrics. And what's really nice is that you can also see the UMAP plots for any given method. And then we have a final section for scalability, mainly scores related to time and memory. So I hope this helps you in deciding which method is more adequate for your integration task. So these are the main findings of this publication, but I will leave some other benchmarking studies in the blog post in case you'd like to do further reading. And of course, I really encourage you to read the actual paper. I'm just summarizing some of the main points here. And I should mention that Lucan's benchmarking study was published in 2022, so it might be missing some newer tools. So yeah, hopefully this will make your life much easier when trying to decide on an integration method. And that is it for today. Squid-tastic. Hope you like this content. Let me know if you'd like to see more videos like this where I go over a particular publication. And if you'd like to see a tutorial on any of these methods, let me know in the comments section down below. Thank you very much for watching. Have a squid-tastic day and see you in the next one.